Hi everyone, my name is Haley and this is my RenR690 final project. So as we all know, we are living in a time of anthropogenic climate change, and one of the outcomes of this is deglaciation. Deglaciation may impact freshwater organic carbon cycling, since glaciers have the ability to store organic carbon and then export it into headwater streams when they melt in the summer. This creates a unique glacially fed stream carbon pool with glacial and terrestrial characteristics that could be different than those of non-glacially fed streams, which predominantly only have terrestrial carbon inputs. Organic carbon characteristics are important because they can be assimilated by heterotrophic bacterial communities, which can play important ecosystem roles in freshwater streams, such as food web structuring, as well as increasing carbon dioxide emissions through respiration. Some studies have suggested that glacial carbon is actually more easily assimilated by these microbial communities because it has a unique structure compared to terrestrial carbon. If glacial carbon is indeed important to microbial communities, then a loss of glacial carbon from the system could change microbial community structure and therefore change the roles that they perform in the ecosystem. This brings me to my first research question, which is to assess whether carbon pools in glacially fed rivers display more glacial or terrestrial carbon characteristics, and how this varies with season and with distance from glacier. Secondly, I wanted to investigate how the microbial communities varied with both season and with distance from glacier. And finally, I wanted to investigate whether the characteristics of the stream organic carbon pools led to variation in my microbial communities. In order to achieve my research questions, I collected water samples in 4 glacially fed streams in Banff and Jasper National Parks to characterize them for microbial communities and carbon characteristics. This was done from May to October in 2019 and along a distance gradient from 0 to 100 kilometers away from glaciers. In order to answer my first research question, I conducted a linear discriminant analysis on organic carbon parameters. This plot here is grouped by proximity to glaciers. It was found that sites sampled near the glacier were significantly different than samples collected from sites far away, and that sites near the glacier showed glacial-like characteristics in that they have low molecular weight compounds, low proportions of humic components, and low amounts of overall carbon. Whereas samples that were collected at sites far away had a mixture of glacial carbon and terrestrial carbon characteristics. They were similar to terrestrial carbon in that they had a high proportion of high molecular weight compounds, and they were similar to glacier carbon in that they had a low amount of humic components and a low amount of overall carbon. This is another linear discriminant analysis of organic carbon characteristics, this time grouped by month sampled. It was found that samples taken in May and June were significantly different than those taken in July, August, or October. Samples taken in May and June had high amounts of aromatic components, which was similar to ranges found in non-glacially fed stream terrestrial carbon pools, and sites later in the year had low amounts of aromatic components, which were more similar to a glacial carbon source. Overall, each site, no matter what month they were sampled in, showed a mixture of glacial carbon characteristics and terrestrial carbon characteristics because, like glacial carbon, they all had relatively low amounts of organic carbon and low amounts of humic components, but like terrestrial carbon, they had high amounts of high molecular weight compounds. To answer my second research question, I performed NMDS plots on microbial communities collected in 2019. This NMDS plot is grouped by the proximity to glacier, and it was found that sites that were taken far away from glaciers were significantly different than sites that were located near glaciers, with the distance range accounting for 6% of the total variation between microbial communities. This is a second NMDS plot, colored this time by the month that the sample was collected. Samples that were collected in May were found to be significantly different than those collected at other times of the year, and overall, the month sampled accounted for 12% of the total variation between microbial communities. In order to answer my third research question, I looked at how my organic carbon parameters might be explaining variance in microbial communities through a distance-space redundancy analysis. It was found that my organic carbon parameters did not significantly account for variation seen in the microbial communities. The conclusion to my first research question was that carbon pools located in glacially fed rivers displayed a mixture of both glacial carbon characteristics and terrestrial carbon characteristics. They had on average low amounts of organic carbon and low humic components like a glacial sample, and had low amounts of low molecular weight compounds and high aromatic components on average similar to a terrestrial carbon or a non-glacial source. The conclusion to my second research question was that season did significantly account for variation in the microbial communities, with month sampled accounting for 12% of the variation seen between communities. Additionally, distance from glacier was also causing significant variation in the microbial communities, accounting for 6% of the variation observed between microbial communities. Finally, the conclusion to my third research question was that the organic carbon characteristics did not significantly explain microbial community variation. Thank you for listening to my presentation. Some references are in the description along with my email if you have any questions.